I know it was.
Shall we continue our discussion? Okay, welcome back. Did anyone have any thoughts during the break? I mean, anything you'd want to share or any questions that you have? Okay, one just thing I want to mention with this distinction between gluconeogenic amino acids and ketogenic amino acids is that they're not mutually exclusive. So some amino acids like tryptophan can be both gluconeogenic and ketogenic because it's got a lot of carbon and they can be split into different things. So it's not, yes, there is some overlap. For example, the branched chain amino acids, some of them can be both ketogenic and gluconeogenic. Okay, but yeah, don't worry so much about memorizing them, I'd say, but just get that concept. And if you're asked if an amino acid degrades into acetyl-CoA, that means one thing, and if it doesn't degrade into acetyl-CoA, that means a different thing. Is that fair enough? Okay. All right, I'm going to have to carefully review my questions now to make sure that that I'm true to that. Okay, so we're going to talk about the degradation pathways for some, not every one of the amino acids, but we're going to focus on branched chain amino acid degradation and amino acids that go through tyrosine, so phenylalanine, tyrosine, and down the line from there. We're also going to talk a little bit about cysteine and cysteine, and not so much a disorder of synthesis, but of reuptake in the kidney. Okay, so let's consider those branched chain amino acids first. So that would include valine and isoleucine and leucine here. So isoleucine is also there, and I think that's it. Okay. So the branched chain amino acids are considered kind of lumped together because, let's see, they're all essential in our diet, so we need to eat them. They're very important sources of fuel in the fasted state within muscle cells. So if you, so if you, for example, drew blood from the vein that's draining skeletal muscle when you're in the fasted state, that blood is going to be depleted in branched chain amino acids, and it's going to be increased in glutamine and alanine relative to the amino acid concentration of the protein in that skeletal muscle. Does that make sense? So basically, in that fasted state catabolism of protein in the muscle, the protein is breaking down the carbon of those branched chain amino acids to carbon dioxide. The nitrogen is being transported to pyruvate to make alanine, or to alpha-ketoglutarate to make glutamate, and then glutamine. Then those amino acids leave and transport the toxic nitrogen away from the muscle cell. Okay, so that's an important role of that branched chain amino acid catabolism in fasted state metabolism. Okay, so in terms of how they get there, the first step is a transamination reaction using the alanine as alanine pyruvate as the acceptor donor system in the transamination reaction. This then leaves an alpha keto acid. So this nitrogen, this becomes a ketone, and it's next to the carboxylic acid. So that's where the name alpha keto acid comes in. So these are all substrates for a enzyme called the branched chain keto acid dehydrogenase complex. And this has a very familiar enzymatic activity and a familiar subunit composition and a familiar vitamin-derived cofactor requirement, as two other enzymes that we've seen before, the pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase in the TCA cycle. So these are all performing an oxidative decarboxylation. So they're making a carboxylic acid into carbon dioxide, and then they're transferring the remaining carbon onto coenzyme A. Okay, so the basic activity is the same as the pyruvate dehydrogenase reaction, and it's using thymine pyrophosphate for the decarboxylation part of it, and then lipoate and coenzyme A for the acyl transfer, and then FAD and NADH as redox cofactors. So that's kind of a common, evolutionarily conserved architecture of those decarboxylation reactions that we've seen before. Okay, so that is important, and it's important, I think, because it's repeated in a couple other contexts that are important in fuel oxidation. It's also an important pathological step. So the inactivation of this step results in a condition called maple syrup urine disease. That's an inborn error of metabolism. The overactivation of that step results in a familiar seizure disorder and like an autism condition. Okay, and then what happens below that, I think, is less important because it's less pathologically significant. But just know that we can be both gluconeogenic and ketoneogenic processes from these amino acids. The valine is gluconeogenic only. The isoleucine and leucine are both ketogenic and gluconeogenic. Okay, so let's consider the degradation of phenylalanine, and this is also the synthesis of tyrosine. So the phenylalanine hydroxylase enzyme, this is adding an OH group from molecular oxygen and using that tetrahydrobiopterin as a redox electron donor, and converting the phenylalanine into tyrosine. So this pathway and that phenylalanine hydroxylase step, this is both important for the synthesis of tyrosine, and tyrosine then can be used for the synthesis of protein, just using tyrosine as a amino acid in translation and bound to a tRNA and all that. Tyrosine is also an important precursor for the hormone, for different hormone synthesis. So tyrosine is a precursor for dopamine and then can be converted into epinephrine and norepinephrine. Tyrosine is also a precursor for melanin biosynthesis. So it's kind of got a few different roles beyond just serving as a protein. Let's see, what else does tyrosine do? Thyroid hormone is made from tyrosine as well. Okay, so that's some other metabolic fates of tyrosine, but if we were going to catabolize it down to the simple parts that we could make fuel from, the first reaction, as with many of these amino acid catabolic reactions, is a transamination reaction. So using the alanine pyruvate system in as the transamination partners. This makes a keto acid, the hydroxyphenyl pyruvate. This carboxylic acid then can be decarboxylated and leave as carbon dioxide to make homogentisate. Homogentisate oxidase then can cleave the ring structure And then further reactions can generate fumarate and then acetoacetate. So the acetoacetate, this is the ketone body, and this can generate 2 molecules of acetyl-CoA. Fumarate then, this is a TCA cycle intermediate, and this is 4 carbons that could be converted readily into the 4 carbons of malate. So this is an example of the same amino acid being both ketogenic and gluconeogenic in the same pathway. Okay. And maple syrup urine disease is important because of inborn errors of metabolism. The phenylalanine tyrosine pathway, that's important because there are several inborn errors of metabolism. PKU is kind of the more common one, the deficiency in the phenylalanine hydroxylase. Then below phenylalanine hydroxylase, there are conditions collectively referred to as tyrosinemias, which, as their name suggests, result in the buildup of tyrosine. Okay, let's look at some disorders, and the first disorder that we'll consider is illustrated in this question. And this is kind of a oddball, I guess, from the way that I just described these conditions. Okay, so this one, we haven't seen anything like this, and I don't think we've seen any of the different distractors until today, but it's really right in the question, right? The cystine in his urine is elevated, so it's probably cystinuria, right? Okay. Okay, good. 100%. So what is cystinuria? So this is an example of inborn error of metabolism that is not involved in a failure of synthesis or of degradation of an amino acid, but is a failure of transport of an amino acid. So I'm just giving it to you as an example, and it's going to get into kidney function, and kidney physiology is kind of bewildering to me, and you're going to get a lot of that in year 2. So I just want to introduce this idea. So what is cystine? We know what cysteine is. That's the amino acid that has the carbon chain and then the free sulfur group, the sulfhydryl group, as the terminus of the side chain. So we know that when cysteine is present in glutathione, that sulfur can either be in the reduced or the oxidized state, depending on whether it's bound to another molecule of glutathione. Remember that? Okay, so the same thing can happen with just the free cysteine amino acid. So the cysteine is when that sulfur group or that sulfur atom is in the reduced form, it can be oxidized to cysteine by making a disulfide bond with another molecule of cysteine. Okay, so there's just equilibrium between the reduced and the oxidized form depending on the redox environment within the body. Okay, I guess the consequence for the pathology of cystinuria is that the cysteine is more hydrophobic or less soluble than the cysteine. Okay, so this is where we get into kidney physiology, and I think, this paragraph here is that's like everything that I know about the kidney. But basically, it's a filter. Your blood goes into it, and then a lot of stuff gets taken out of the blood and goes into a tube that ultimately becomes the urine. But before it goes into the urinary bladder, it goes through a big loop and stuff gets filtered out of it and back into the blood. So we kind of filter out a lot of stuff, and then good things or things that are useful like amino acids, those can get recaptured. So that's the recapture part is shown here. Augmentate. So this is the lumen of the duct that would become the urine. Okay, and then these are the cells that are trying to, get anything that's useful that we don't want to pee out and get that back into the blood. So there are a host of amino acid transporters, and they're shown here, and there are inborn errors of metabolism that result in over peeing out, if you will. I'm not sure if that's a medical term, but increased urinary excretion of different amino acids, depending on which of these transporters is defective. Now, the cystinuria, that is resulting from a deficiency in either of two subunits of the transporter for the oxidized form of cysteine. And the problem here isn't necessarily that you are peeing out too much cysteine or losing cysteine. It's that the cysteine is less soluble, and that can precipitate out and make a stone in this duct, and then you've got failure to filter out stuff into the urine, and when that gets occluded. Okay, is that clear? All right, so you're going to learn a ton more about the basic physiology and a whole host of other kind of similar conditions, but I just wanted to put that in there because it's a nice contrast to some of the other conditions. Now let's look at this kind of same question, or similar question here. Or similar So one feature of amino acid transporters is that they tend to not be specific for one amino acid, but they'll transport classes of amino acids or chemically similar amino acids. So that's why we've got the arginine and lysine and ornithine are also elevated in the urine, because they're using that similar transport system. But if he's peeing out more cysteine, why is cysteine in his blood normal? Yeah, very good. So cysteine is not an essential amino acid. We don't need to eat it in our diet. We can synthesize it, and if we have a peeing out too much of it problem, we can just increase the rate of synthesis. But that would depend on which other amino acid. Okay, so the only other sulfur-containing amino acid is, of course, methionine. Okay, good. All right, so let's look at some disorders further disorders of amino acid catabolism that aren't related directly to these transporters. And we'll talk about the phenylalanine tyrosine pathway with PKU and the tyrosinemias, and then the branched chain amino acid catabolism disorders and autism like condition with overactivation of the branched chain keto acid dehydrogenase, and then maple syrup urine disease with the inactivation. Okay, so let's look at maple syrup urine disease. Maple syrup urine disease is resulting from inherited loss of function mutations in that branched chain keto acid dehydrogenase enzyme. And the presentation is as a neurological disorder. The main name of it comes from a characteristic sweet smell in the urine that occurs right soon after birth, and that's a result of these alpha keto acids accumulating and degradation products of them being excreted into the urine, and they give it a characteristic sweet smell because of, and hence the name of the condition. So this is a disorder that is, it's screened for with metabolic screens in most states, so luckily, or hopefully it would be identified before it would be symptomatic with the typical screens for metabolic disorders. If it wasn't identified there and was allowed to progress naturally, you would get that sweet smell in the urine, and then you would get neurological symptoms, and if it wasn't corrected, they could manifest in seizures and cognitive deficits and like mental retardation. Because the branched chain amino acids are such important sources of fuel in the fasted states, if their catabolism is compromised, you can get a compensation with for increased glycolysis that can result in lactic acidosis and hypoglycemia, and that starvation could also manifest as increased fatty acid catabolism and a ketone body response. So the treatment for this condition, acutely you want to dilute out the keto acids that are that are problematic, so hydration and maybe a what do you call that, dialysis. So that would be if you're having an acute crisis. The condition can be managed relatively okay with a diet where the branched chain amino acids are very limited. So this is, kind of a, if you think about managing this, it would be challenging because any kind of a protein you had, you would have to be very aware of its amino acid content, and you have basically a no protein diet, and then all of the protein that you have is synthetic protein that's depleted of the branched chain amino acids. Okay, so there's different presentations of this condition. The classical maple syrup urine disease is basically the presentation is shown here, where it's a very sudden onset after birth, and the symptoms are pretty severe, and this is due to complete loss of activity of this enzyme. So the enzyme is completely dead. So let's consider a couple of questions here. And I guess I kind of led into this in our discussion earlier. But if you remember the E1 subunit, that's responsible for doing that decarboxylation step, the first step in the pyruvate dehydrogenase reaction. And that is using thiamine pyrophosphate. Very good. So same question about branched chain keto acid dehydrogenase is the same answer here. Thiamine pyrophosphate. Very good. So that gets into treatment options for people who have the classic or the mild, or excuse me, the non-classic maple syrup urine disease. And this can be either a mild presentation where the symptoms aren't as severe, or it can be an intermittent presentation where the symptoms come and go. And with this intermittent presentation, that's frequently related to high protein meals. So if you have a high protein meal, you're going to have a higher dose of these branched chain amino acids, and then you're going to have problems when you try to catabolize them. So the difference between the mutations that result in the classic and the intermittent and mild forms of the maple syrup urine disease is that the classic form, the mutations cause a complete loss of the enzyme activity, whereas the mutations that result in the intermittent or the mild presentations, there's still some residual enzyme activity. And that gets at a treatment for people who have the intermittent and mild forms. They can be treated with oral thiamine, the vitamin, and the idea there is that you're trying to maximize what residual activity you have by making sure that the enzyme has as much cofactor as it possibly can have. Okay, so these guys have a little bit of that keto acid dehydrogenase activity left, so you never want to make lack of cofactor a limiting factor on what activity you have, is kind of the idea there. Sound square? Okay. Okay, so let's look further at that keto acid dehydrogenase complex and its similarities with the pyruvate dehydrogenase complex. Okay, everybody get a chance to answer? Ooh, very good. So phosphorylation or dephosphorylation, it's got to be one of those two, right? In the case of the pyruvate dehydrogenase complex, there's a family of pyruvate dehydrogenase kinases that phosphorylate and inactivate the enzyme. Right? And then which type of enzyme activates it then? That is the phosphatase enzyme. Okay, good, phosphatase enzyme. So pyruvate dehydrogenase, it's regulated by reversible phosphorylation. So in conditions where we have abundant energy, for example, lots of NADH around, lots of acetyl-CoA around, there we want to inactivate it and kind of choke off the energy generating pathway. Yeah, not shown here, hypoxia is another thing that will activate those kinases. Then calcium, which is a kind of a signal of work being done, that will activate phosphatases, which will dephosphorylate the pyruvate dehydrogenase and activate it, and then it does its job of turning pyruvate into acetyl-CoA and providing a substrate for the TCA cycle. Okay, so why are we going back to the pyruvate dehydrogenase complex? Well, the regulation of the branched chain keto acid dehydrogenase complex is recapitulating the pyruvate dehydrogenase complex, and that is that it's being regulated by reversible phosphorylation. And this has pathological consequences that were identified, first published in 2012, looking at a pedigree analysis of families that had inherited autism-like condition, but it was not a typical autism. It was autism plus a seizure disorder. So it's not like this isn't accounting for most conditions or most autism. This is a very rare form of autism. So using whole exome sequencing of the members of this family that had the two affected individuals that had this autism and seizure disorder, they found that in the wild type sequence of a gene is shown up here, and there was a G to C mutation that resulted in a arginine to proline mutation, and arginine to proline, that's non-conservative, of course, substitution. And it was, these two affected individuals were homozygous for it. Okay. And what was that gene? That gene was branch chain keto acid dehydrogenase kinase. Okay, so that is the gene or the enzyme that would normally be phosphorylating and inactivating the branch chain keto acid dehydrogenase. So in the patients that had this autism slash seizure disorder, they had essentially unregulated catabolism of their branch chain amino acids. Okay, so normally the activity of the branch chain keto acid dehydrogenase kinase, this would maintain some equilibrium of the branch chain amino acids and allow them to be available for protein synthesis, for example. But in the patients with this condition, that regulation was totally absent. Okay, so there was a mouse model that was generated that had the same mutation in the corresponding mouse gene. They were fed a normal diet or a diet that had a normal amount of branch chain amino acids in it. And wild type mice, they didn't have any of the seizure or autism-like behaviors. The knockout mice, days after birth or days after weaning, excuse me, when they're on that diet, they started to exhibit the signs of mouse autism, essentially. So that's kind of aggregated over here. And it's broken out into seizures and hind limb clasping. It's like a just repetitive motion that mice do that's, you know, somewhat redolent of autistic behavior. Okay, so this is demonstrating that that mutation is causative for similar deficits in an animal model. What's most significant, I think, is that it's potentially a treatable condition. So this is showing, again, on the y-axis, this is the number of these, you know, abnormal or autism-like behaviors on each day. Looking at mice that were on their normal diet here, and then they were on a high branch chain amino acid diet over here, and you can see that those adverse activities start to taper off. So this was, you know, pretty significant. Again, not that this is representing most people with autism, certainly, but autism is kind of refractory to a lot of treatments in a lot of cases. So this is representing a potentially treatable form of autism and the dietary intervention that's, you know, fairly simple. So yeah, I've tried to follow up on this. I mean, it's super rare, so the publications aren't super frequent, but some of the recent research is suggesting it's not just a problem with the depletion of these branch chain amino acids, but that this enzyme, when it's running at full speed with no regulation, that it's generating oxidative stress, and that's kind of contributing to the pathology. So that would maybe, you know, argue against having a high branch chain amino acid in the diet. Okay. Any questions on that? Yes.
How is eating more of that? Because it looks downstream of the lab. Am I?
Yeah. So the initial idea was that in the people and in the animal model that the adverse events were due to starvation of tissues for branched chain amino acids. They either needed it for energy or they needed it for synthesizing proteins that were essential for neuronal function. Okay, so that was kind of their idea and rationale behind that. But this newer data, or newer, I don't know when, 10 years ago or 8 years ago, would suggest that that's not a good strategy. So they are trying this diet and have tried it in the families that have this condition, but I haven't seen that it's been published, the clinical outcomes of it. But I just thought it was kind of interesting and just illustrating the different consequences of inactivation of that enzyme complex and then overactivation of the complex. Okay, so let's change gears and talk about disorders of phenylalanine metabolism. So phenylalanine metabolism, inherited mutations involved in the enzymes within this pathway will result in phenylketonuria with a deficiency in phenylalanine hydroxylase. And then downstream of that, we have different tyrosinemias. And then alkaptonuria, it's named alkaptonuria, but it's lumped in with the tyrosinemias. So let's consider PKU, or phenylketonuria, first. So the reaction, the relevant reaction, is the addition of an OH group onto the ring structure of tyrosine. Phenylalanine hydroxylase is the enzyme that does this, and the redox cofactor is tetrahydrobiopterin. So tetrahydrobiopterin, this can either be in the reduced or the oxidized state. The enzyme that will reduce the tetrahydrobiopterin and make it available for the phenylalanine hydroxylase is dihydropteridine reductase. The classical phenylketonuria is caused by mutations in the phenylalanine hydroxylase gene. There's a non-classical PKU that can result in mutations in the dihydropteridine reductase enzyme. So if you can't make this cofactor, or if you can't regenerate it, then you get recapitulate the symptoms of PKU. And again, this cofactor, this tetrahydrobiopterin, this doesn't have a vitamin precursor, so we can synthesize this fairly simply. Okay, so PKU, the symptoms like maple syrup urine disease are associated with or manifest shortly after birth. And like maple syrup urine disease, there's a characteristic odor associated with it. In this case, it's frequently referred to as a mousy odor, and this is coming from the phenylacetate that is the alternative metabolic product of the phenylalanine metabolism if it can't go down this pathway. So the mousy odor comes from the phenylalanine being degraded into a different pathway. There is also frequently a light complexion with people with PKU, and this is reflective of the role of tyrosine in synthesizing melanin, that pigment melanin. They can also then, the other symptoms would be more neuronal, so seizures and cognitive delays. This is presumably resulting from tyrosine's role in synthesizing dopamine and downstream neurotransmitters. Okay. Oh, and so the diagnosis, generally if a baby is born in a hospital setting in the United States, you would diagnose this with metabolic screening shortly after birth, and it would be identified, and you could get an intervention before you would get any of these symptoms. The intervention is to have a phenylalanine restricted diet. So just only a tiny amount of phenylalanine that you would need for protein synthesis. and not so much that you would be building up the toxic intermediates. Tyrosine then would also become an essential amino acid, but you don't. Tyrosine is not. If you're eating protein, you'll get some tyrosine, I guess. Okay. Let's see. Okay, so the degradation of phenylalanine, if it can't go through this hydroxylase reaction, the phenylalanine is transaminated, and then it becomes the phenylacetate. And yes.
You're restricting phenylalanine to treat this disorder as the main cause of accumulation of phenylalanine or lack of tyrosine?
It's kind of both, and it's not. So you have to have like a synthetic protein diet, or you have to watch your protein and make sure that you're not getting phenylalanine. Protein is also where you would get tyrosine, so you gotta know how much tyrosine you're getting. But the issue isn't so much of how much you're putting into your mouth as it is with transport. So remember that I mentioned earlier that amino acid transporters tend not to be specific for one amino acid, but they're kind of family, or one transporter will transport families of amino acids. Okay, so that's a great segue into this next slide, which there was just a new review that came out on PKU last year, and that this is a good illustration of it. So there is a transporter that transports tyrosine, but it also transports other large neutral amino acids, or LNAs. So and that would include tyrosine and tryptophan and phenylalanine. So in the case of a PKU, you would have elevated phenylalanine, and this would be in the untreated. Say you're eating normal amounts of phenylalanine. This elevated amount of phenylalanine in the blood, this is going to act as a competitive inhibitor for the transport of those other amino acids, like tyrosine and tryptophan. Okay, because this transporter, it has that overlapping specificity if there's just only phenylalanine around, or if it's around at way higher concentrations than the others are never going to get transported out, or excuse me, transported through that transporter. So that results in a decrease of not just tyrosine in the brain, but also of tryptophan. And so the pathology of the PKU then is the neuronal symptoms are thought to be due to a lack of serotonin synthesis from the tryptophan, and then a lack of dopamine from the tyrosine. And this could be, even if you had plenty of tyrosine that you were eating, you could still get this problem with intracellular transport if you had way too much phenylalanine around. Is that clear? Does that help? Okay. Okay. Are there other questions? Okay. Let's see. This dashed line here, this is I think meant to illustrate a speculative inhibition of the phenylalanine on those enzymes that are using or that are involved in converting tyrosine into dopamine and tryptophan into serotonin, respectively. So again, just if that pans out, exacerbating the issue. Okay, and then I think I mentioned this earlier, but yeah, if we get a inherited loss of function mutations in the dihydropteridine reductase enzyme, that can recapitulate the symptoms of PKU. Okay, so let's consider next the tyrosinemias. So tyrosinemia, that's going to be anything that would result in an increased levels of tyrosine in the blood. And there are several mutations that can result in the tyrosiniemias, and I'll just cover them from north to south. And if we run out of time before the end of class, we'll finish it up on Wednesday. Okay, tyrosinemia type 2. This is a defect in the amino transferase enzyme that's clipping off the amine group from tyrosine to make the hydroxyphenylpyruvate. So the tyrosinemia type 2, this is not the mildest, but one of the milder of the tyrosinemias. So this is can result in mental retardation, but not the severe sudden onset seizures and such associated with PKU or with other tyrosinemias. There is this characteristic plaquing on the feet, and this can also occur or manifest as corneal ulcers. So the treatment, if the tyrosinemia type 2 is identified, is to have a synthetic diet, and here we're not just trying to limit phenylalanine, but you'd have to limit both phenylalanine and tyrosine. Okay, so further down the pathway, tyrosinemia type 3. This is one of the rarer ones and the more severe ones, and this is associated with more severe cognitive symptoms resulting from again, lack of neurotransmitter synthesis in the brain, and then the toxicity of the upstream products here that don't have a way to get down to the simple products. Sorry. Okay, so tyrosinemia type 3, this is rare and severe. Alkaptonuria, the deficiency of homogentisate oxidase, this has the, I guess, distinction of being, or generally considered to be, the first inborn error of metabolism that was described by modern medicine in about early 1900s, I think. So alkaptonuria, this is the deficiency of homogentisate oxidase results in the accumulation of homogentisic acid, and that can, again, this is another case where we can get kidney stones or renal calculi. So we've seen 3 cases so far where we can get that, right? Glycine catabolism through glyoxylate, cystine accumulation through the cystine transporter mutation, and now the alkaptonuria, which makes or results in the buildup of homogentisic acid, and that can then, if that builds up in the lumen of the kidney tubules, then that can precipitate out and result in a stone. Okay, so kind of the hallmark of alkaptonuria is ookernosis, or a buildup again of that same homogentisic acid in the sclera of the eye, and then also in cartilage, and you can see that as a dark spot in the cartilage in the ear. Okay, tyrosinemia type 1 is that's the final tyrosinemia. That is a loss of function mutation in fumarilloacetone hydrolase. The fumarilloacetone hydrolase is the final enzyme that's cleaving the tyrosine into fumarate and acetate. When this enzyme is absent, the succinyl acetone is a toxic intermediate that builds up, and that can cause liver failure. So the tyrosinemia type 1, this is the most severe of the tyrosinemia, and results as a very rapid onset liver failure. And this one has the worst of the outcomes. Nitosinine, this is a treatment for the condition that the mechanism of action is to slow the catabolism of tyrosine and the derivatives and try to limit the buildup of or limit the production of the succinylacetone to an amount that's not toxic or that can be excreted without toxicity. However, there are other things up here that can also be toxic as well. So it's not the greatest treatment, either conceptually or in practice. Okay, are there some questions? Or do you have questions? Okay, I've got a few clicker slides and some clinical cases, a couple clinical cases. I'm going to put all those in a download that will be available on Canvas in just a couple of minutes. And we'll go through those. I think we'll have plenty of time to cover them on Wednesday. But if you've got a chance to look at them ahead of time, then it'll go quicker. Okay, all right, bye everyone.
I was like, in my mind, I was like, formulating how that phrase went because I didn't know. I was like, all right. I didn't.
Oh, you didn't.
So, I know, that's why I was like, I'm not situation. Monday, I thought it was Wednesday. Oh, ground vegetation. I did not do that. Oh, really? Is this just for my ringer?
I don't know, at least I didn't. I mean, I'm planning on using it.
Whatever.
I didn't sign up for it.
What's today? You're right. What time is that? Our next one? The OB. Unless you want to do the first interest.
You guys did that officially, or is that?
This is the official.
I don't know, maybe if you want to stay interested in this for longer, maybe you don't want to go and see what it's really like.
Discernment.
So, you like fill out a form
Right.
today. You're going to give us whatever information.
Don't want to do something, it's better to know.
Right. Right. No, exactly. I feel like knowing what you aren't is more important. Sometimes way more important than knowing what you are. It helps you figure it out. But anyway, really appreciate it.
Maybe it's something.
And she
Just like
said actually liked it. Learned a lot. So. Right.
I was looking at it actually a couple weeks ago.
Yeah.
Yeah, they do.
Yeah.
I know. Yeah. I was telling Matan, I was like, oh shoot, Lauren might actually. Still morning. I was like, why does yours look different, but it's a different edition? Well, it's the Q&A. No.
Annie wants to hear?
It's like questions. Yeah, I'm in second year again. She ordered on Amazon. Took step now. And she said it was good. You pointed in the resource, or she just had it? Didn't use it.
Yeah.
Oh, down there.
Oh, I see your backpack. Right here?
That's fair. But she had like a different like step. Do you think we're gonna grab another sandwich? I feel like normally we can. Oh, you just already went for it.
You should have.
Should we go back? Well, we'll let people go through. Maybe after this before I go study. It's so chilly. Never.
I feel bad. I know.